Assalamualaikum warahmatullahi wabarakatuh, and a very good morning to today we will present about natural product chemistry of Melicope Glabra. Before that, my name is Shazana Hanis binti Abdul Malik and my metric number is 203568. Okay. First of all, we will take a look at Bloom taxonomy of Melicope Glabra. Melicope Glabra belongs to the kingdom Plantae as well as other plants such as trees, herbs, bushes, scrub, ferns, vines, mosses, and algae. This kingdom can be divided into three main divisions, which are Bryophyta, Pteridophyta, and Spermatophyta. Melicope glabra is in the subdivision of Spermatophyta or Magnoliophyta, which is formerly classified as Angiosperm and widely known as flowering plants. This division consists of all seed-bearing plants and can produce a flower. The general characteristic of plants in this division are they have roots, stems, leaf, can undergo photosynthesis since they have chlorophyll, usually undergoing bisexual reproduction, exhibiting double fertilization, and have highly developed vascular tissue, which their xylem has tracheids and vessels, while their phloem has companion cells. This division can be further divided into two classes, which are Liliopsida and Magnoliopsida. Melicope glabra is in the class of Magnoliopsida, which means it is the dicotyledon plant. Plants that are dicotyledon usually have two seeds in their embryos, broad leaves, and have network of veins. Cross-section of stems reveal vascular bundle arranged in rings and have taproot system. Their flowers usually consist of 4 to 5 petals. Next, the order for Melicope glabra is Sapindales. Sapindales consist of woody plants such as trees, shrubs, and woody climbers, which can be further divided into 9 families, which are Anacardiaceae, Bibertheniaceae, Burseraceae, Kirkiaceae, Meliaceae, Mitrariaceae, Sapindaceae, Simorobaceae, and of course, Rutaceae. Melicope glabra is one of the plants in the Rutaceae family. Rutaceae family are usually small to medium shrubs and trees. The leaves are often aromatic, containing oil glands, and their flowers usually have 4 to 5 sepals and petals that are white, yellow, purple, pink, or red in color. There is approximately 160 genus in the Rutaceae family, including Melicope. So, plant in the genus Melicope have simple or trifoliate leaf arranged in opposite pairs or sometimes whorl. The flowers are arranged in panicles with 4 sepals, 4 petals, 4 or 8 stamens, and usually bisexual. There are 4 or 5 carpels fused at the base with fused style, and their stigma are similar to the tip of the style. The fruit is composed of up to 4 follicles fused at the base, each with 1 or 2 seeds. Lastly, Melicope glabra is one of the 230 species of Melicope. Glabra is from the Latin word which means hairless. Melicope glabra grows naturally in Sumatra, Peninsular Malaysia, Singapore, Java, and Borneo. Its habitat is primary forest. It can grow up to 40 meters tall, and the fruits are round to elliptic and can measure up to 0.4 centimeter long. Next, I will pass to the next presenter for the traditional usage of the plant.
Assalamualaikum and hi. My name is Akila Farha binti Abdul Rahim and my metric number is 200709. I will explain about the traditional usage of the plant. Melicope glabra, which is a plant belong to Rutaceae, is among the significant sources of flavonoids and coumarins. It is also commonly called as pepau daun besar or tenggek burung in local Malaysian. The leaves of the plant are consumed for general well-being. In Malaysian traditional use, the plant is effective to treat cough, fever, and also malaria since it contains coumarins in it.
Assalamualaikum and hi, my name is Amirah Batrisha binti Muhammad Badrul. My matrix number is 200214. So I will continue this presentation with the class of second metabolites in Melicope glabra plant. For this plant, the class of second metabolites is phenolic. Phenolic compounds are a group of natural constituents that are able to provide antidiabetic therapy. It is also categorized as flavonoids, lignans, stilbenes, coumarins, and others. So in this plant, there is 9 phenolic compound was identified. So 4 of them are flavonoid. Flavonoid have effects on preventing degenerative disease and are effective for treating diabetes. This was attributed to their structural characteristics such as the double bond between carbon 2 and carbon 3 of ring C and also the catechol structure of ring B. This compound identified as flavonoid as they have fragmentation pattern. Besides flavonoid, there are other phenolic compounds such as cinnamic acid derivatives, glucoside, coumarin, and also lactone. Renifolin was assigned as glucoside due to the breakage of the glycosidic bond. So to conclude, the presence of phenolic compounds are significant to the anti-diabetic effect. So I will pass this presentation to next presenter.
Next, I will explain about the medicinal usage of secondary metabolites. First is flavonoids. Anthocyanidins, flavonols, isoflavones, and flavonols have been suggested as useful supplements to manage and also to prevent diabetes by improving profile glucose metabolism and hormone regulations in the human body. The anti-diabetic efficacy of flavonoids specifically by inhibition of dipeptidyl peptidase 4 and alpha amylase. Secondly is lactone. Lactone tablets used for treatment, control, prevention, and improvement of the following disease, conditions, and symptoms of fluid retention in liver or kidney disease. Secondly is high aldosterone levels, low potassium levels, heart failure, and also for high blood pressure. Lastly is coumarin. Coumarin is used as blood thinner that are used to keep blood flowing smoothly and prevent the formation of the blood clot. Coumarin has been shown to activate other cells of the immune system. In traditional usage, it is to treat malaria.
Hi and Assalamualaikum. My name is Nur Hayatna binti Mufni. My metric number is 200643. I am going to present about the biosynthesis pathway of 3 of the secondary metabolites found from Melicope Glabra species. First is the biosynthesis pathway of methyl 3,4,5-trimethylcinnamate. Secondly is the biosynthesis pathway of isoramnitine. Lastly is the biosynthesis pathway of trans-ticrosidenol. So first to start is methyl 3,4,5-trimethylcinnamate. To briefly explain, methyl 3,4,5-trimethoxycinnamate is an ester derived from cinnamic acid, where the precursor is 3,4,5-trimethoxycinnamic acid. Okay, so there are 3 methoxy group attached to the phenyl group in the structure of trimethoxycinnamic acid. For the biosynthesis pathway of the methyl 3,4,5-trimethoxycinnamate, cinnamic acid is biosynthetically produced from shikimic acid pathway, where shikimic acid is synthesized by the precursors, which are the phosphoenolpyruvate reacts with the erythrose 4-phosphate and then produce shikimic acid. Shikimic acid then be converted into shikimic 3-phosphate under the action of enzyme shikimic kinase, and then shikimic 3-phosphate further react with phosphoenolpyruvate PEP to produce 5-enolpyruvyl shikimic acid 3-phosphate. And then this compound is further converted into coracetic acid under the enzyme coracetic synthase. Coracetic acid, and then further converted into perfenic acid, and then into L-arginate. L-arginate with the amine group located at the carbon next to carbon COOH group. Okay. L-arginate then be converted into phenylpyruvic acid, and lastly to phenylalanine. Phenylalanine is an essential compound in the shikimic acid pathway to produce cinnamic acid. So cinnamic acid, it has phenyl group and 3 carbon chain in the molecule. So after trans-cinnamic acid has been synthesized by this pathway, so it can be further derived into its derivatives, which is 3,4,5-trimethoxycinnamic acid, and then methyl 3,4,5-trimethoxycinnamate is synthesized. Okay, so this is the whole pathway. Next is the biosynthesis pathway of isoramnitine. Briefly explain, isoramnitine has the synonym of 3'-methoxyquercetin. So from the name, we can know that it is a derivative of quercetin, where this compound quercetin is a flavonoid. Isoramnitine or 3'-methoxyquercetin is a flavonoid, specifically is a monomethoxyflavone. So if you compare between these two structures, between quercetin and isoramnitine, there is a methoxy group attached to the carbon number 3' in isoramnitine, where in quercetin there is no methoxy group. All right. So quercetin is also biosynthetically produced from shikimic acid pathway. So same as before, shikimic acid is synthesized by the same precursors, and then to shikimic 3-phosphate to 5-enol-pyruvate shikimic acid 3-phosphate, corysmic acid, and then into prefenic acid. So after prefenic acid has been synthesized, it can go, it can undergo a number of different pathways to synthesize para-coumarol coenzyme A. So this compound para-coumarol coenzyme A is essential for the synthesis of flavonoid quercetin. Okay. For example, here we can take one pathway from prefenic acid. It be converted into L-aragonate and then into phenylalanine, into cinnamic acid, and then next into para-coumaric acid, and then lastly into para-coumarol coenzyme A. Okay. In para-coumarol coenzyme A, there is a hydroxyl group at carbon number 4. That's why it is named para-coumarol coenzyme A, and also coenzyme A structure located at the carboxyl group, next to the carboxyl group. And then after this compound has been synthesized, naringenin chalcone synthase enzyme converts para-coumarol coenzyme A into naringenin chalcone. So now the pathway has entered the flavonoid pathway. Okay. Naringenin chalcone next is converted into naringenin via the action of enzyme chalcone isomerase. So in naringenin, there is 3 ring structure. And then flavanone 3-dioxygenase converts naringenin to dihydrocamphorol. So there is new OH group in the dihydrocamphorol structure, and then dihydrocamphorol be converted into camphorol, and lastly to quercetin. Quercetin can then be converted into isoramnitine. So this is the whole biosynthesis pathway of isoramnitine which go through the flavonoid biosynthesis. Starts from para-coumarol coenzyme A. And lastly is the biosynthesis pathway of trans-tickersidinol. So briefly explain, trans-tickersidinol is a coumarin, specifically is a pyranol coumarin, which has a 3-ring structure. So trans-tickersidinol is derived from umbelliferol, which is also derived from shikimic acid pathway. So trans-tickersidinol is a dihydroxantiletine type of coumarin. So if we compare between xantiletine and trans-tickersidinol, there is two new hydroxyl group attached to the ring structure of the trans-tickersidinol. So that's why the group is named dihydroxantiletine. So same as before, there is phenylalanine biosynthesis. After phenylalanine has been synthesized, it undergoes conversion into cinnamic acid, and then also into para-coumaric acid. Para-coumaric acid also can be derived from tyrosine, same as before. This one, para-coumaric acid can be derived from tyrosine, also can be derived from phenylalanine. Okay. After para-coumaric acid, and then this compound is converted into para-coumarol coenzyme A, and then the next step is an important one because 2,4-dihydroxycinnamol is synthesized from para-coumarol coenzyme A under the action of enzyme transfer-coumarol coenzyme A to hydroxylase. So we can see new hydroxy group replace proton here. So next is 2,4-dihydroxycinnamol converted into umbelliferol. Now it has 2 ring structure and further going through the third ring formation to form trans-nickel-sidenol umbelliferon, from umbelliferon to D-methylsuberothene to xanthillatine, and lastly to trans-nickel-sidenol. So that's all from the biosynthesis pathway of these three compounds. Overall, trans-nickel-sidenol, isoramnitine, and methyl TMCA all has same type of pathway, which is the shikimic acid pathway. But all these three compounds has different specific pathway that they going through, they undergo. And of course, other secondary metabolites such as lactone group and also glucoside group has other type of different pathway. So that's all from us. Thank you so much.